The overall goal of this procedure is to use a custom-engineered zinc-finger nuclease, or ZFN, to introduce a disruptive mutation at a specific locus to ultimately create a knockout cell line. This is accomplished by first analyzing the DNA sequence of the target gene with the CompoZr algorithm to generate in silico ZFN designs. The ZFNs are assembled, delivered to cells by nucleofection, and screened for activity at their respective gene target sites. For generating a knockout cell line, the cell type of interest is first treated with an active ZFN. Then this population is segregated to single cells, and finally these clonal populations are expanded for additional genotypic characterization. Clones with disruptive non-homologous end joining events at the target site may be studied further in the context of functional gene knockout. Ultimately, genetic characterization, including the cell 1 assay and DNA sequencing, are necessary to confirm observed changes in cellular phenotype as a result of ZFN-mediated genetic modification.
The main advantage with zinc-finger nucleases relative to existing methods, like traditional homologous recombination, is CompoZr ZFN-enabled gene editing allows researchers to create specific and predictable genetic modifications without selection. This includes gene knockouts, gene modifications, and gene integrations.
The ZFN design process begins with simplified bioinformatics studies, which focus on identifying unique regions of sequence containing sufficient number and quality of zinc finger binding motifs that are conducive to performing gene knockout. A clearly stated experimental strategy will greatly facilitate this process, as will information on the target gene, such as atypical